So Eva, take a mic and give the introductions to the speaker. Thank you.
So, Jakub, thank you very much for the kind introduction. And maybe I will just give you a word of explanation, because actually this session was supposed to be led by Anna Wach, who is one of the organizers of this workshop. But unfortunately, due to the health reasons, he cannot join us today, and I will lead the session in her place. So in this session, we will have 4 presentations. The first part consists of 3 talks, and we will get an overview of research potential of nano and micro X-ray spectroscopy beamlines. And this part will be followed by a talk with the aim to provide us with some information on Ukrainian Synchrotron Association. So maybe one remark concerning the questions. So we have 2 options how to ask a question. So just wait until the end of presentation and ask the question, or you can write a question in a chat anytime you like. And afterwards, I'm going to read all the questions if we have enough time. If not, I will ask the speaker to answer the question. So let's start. So the first presentation will be given by Małgorzata Makowska, who is beamline scientist at microXAS beamline at Swiss Light Source, and also material scientist at Advanced Nuclear Materials Group at Paul Scherrer Institute in Switzerland. So, and the title of her presentation is Chemical Imaging and at the microXAS beamline and Plans for the Bright Future at microXAS 2. So Małgorzata, the stage is yours.
Okay, thank you. And first of all, thank you for the invitation. For inviting me here. So let me just share the screen. So again, also in my case, can you confirm that you see the presentation mode?
Yes, we can see your presentation.
Okay, so thank you again. That's one of rare occasions when I don't need to repeat my name. So thank you for that, for the introduction. So yeah, I will present you some selected, a little bit description of our beamline microXAS, which is located at SLS, Swiss Light Source here, as you see on the picture. And but I will focus also on scientific cases that we covered during last years. And then I will tell you also about our plans about the upgrade of the beamline, so new possibilities for the future. So our team, so this is our team, the beamline. We are 3 beamline scientists. Daniel Brodimont is our group leader. Then the second scientist is Dario and me, myself. We have a great support of 2 technicians, Beat and Mario, and we have 1 postdoc currently working with us. So, as I said, I will introduce you a little bit the beamline layout, and then I will focus on some examples of science cases that we tackle. So these are actually 4 main, most frequent scientific directions that we work on recently, but also that we aim to force in the future forward. So I will show you some examples about radioactive materials, because here I would like to mention that microXAS is the only beamline that at SLS, at the synchrotron, that has a license for studies of radioactive materials. And then we also measure some biological samples. We do quite a lot of studies on electrochemical devices like lithium-ion batteries or other types of batteries, but also electrochemical cells like fuel cells or electrolysis cells. And since a couple of years, we spend a lot of time also on experiments on advanced manufacturing, in particular additive manufacturing with lasers. but it's a bit broader than just a 3D printing. And then, as I said, I will introduce you plans for the future. And in the end, if I still have time, I would also like to give some examples from Phoenix beamline, which is actually maybe a more interesting case, as microXAS is operating at a bit higher energies usually than we are discussing today. And Phoenix fits a bit better. So our main, the main goal or activity at microXAS beamline is chemical imaging, and that's also how we call our group. It's a chemical imaging group. So what is the difference between physical imaging and chemical imaging? So physical imaging, that's at least our understanding of it. This is imaging that gives you information mainly about morphology, structure, and it's with X-rays, it's typically based on absorption contrast and phase contrast. What we do at microXAS, we provide images so we can see structures with spatial resolution based on a chemical contrast, so chemical composition. We can see distribution of different elements, but also with spatial resolution, we can visualize speciation of or chemical states of specific elements. And here's an example, that's our example case, what we did. So this is an image done with optical microscope and with SEM of some kind of writing. And this is an example of physical imaging where you see the morphology, the surface structure. But if you do the chemical imaging, then you can kind of deconvolute the information there. And this is, of course, an example of fluorescence contrast mapping, where we could see that the fluorescence of silver and fluorescence of gold gave us a different image, and we could read what's written. So the beamline, as it was in the past up to last October, looked a little bit like this. So this is a simplified sketch of the beamline. So our source was undulator U19. So one of these undulators that Luke was talking about, that perhaps will travel to Poland, perhaps the other one. Then in the optical hatch, the two main components were one mirror, which a dynamic focus in one direction and collimation option in other direction. Then we had the fixed exit monochromator, and in the hatch, experimental hatch, we still have two sets of KB mirrors for two different sizes of focus. So we could operate with focusing at around 10 microns or at 1 micron. And at the second station where we would get this 1 micron, we would lose a little bit of flux because of the acceptance of the mirrors. And then also we had options to do the focus with gratings, but also sometimes we used the full field microscope, so we could actually use perform the full field imaging and relatively easily switch between focused beam and parallel beam, collimated beam for the full field imaging. So that was quite an interesting and unique option. So up to now, we were operating in this range of energy from 3 to 22 kVs. However, in reality, it was rather between 4 and 20. So that was like a reusable flux. At the edges, we had very few photons. And this is a sketch of how the final sample position looks like. So we had the always a manipulator. So that would be a case for some ex situ experiments where we have a manipulator for the sample, which can be moved, which can move a sample in all the direction, X, Y, Z, with also with a rotation and with a swivel. So we could also correct for the alignment. Simultaneously, we were able to measure fluorescence and diffraction from the sample during the scanning over the sample, and also transmission signal through the sample. So these three signals we could measure at the same time using monochromatic beam. And of course, another type of measurement that we used to do as a standard measurement was absorption spectroscopy, XANES or XAFs. But then this, of course, had to be done with energy scan, so not simultaneously with the others. But it could be done at the same sample after selecting a specific point, for instance, based on diffraction and fluorescence images. So now I will present you some science cases. So as you heard, as I was introduced by Eva, she mentioned that I also work in the nuclear materials group. So several examples I will present you from this field. So what are the challenges in nuclear fission reactors? So here we have the challenges that we tackle at microXAS. So this is maybe something new for you, new here. So this image shows a pellet of spent nuclear fuel. So this is essentially uranium oxide embedded or placed in a cladding made of zircaloy. So here we have, during the operation of the reactor, we observe a number of structural changes inside the pellet, inside the uranium oxide, but also we have precipitation of hydrides in the zircaloy claddings. We have oxide formation at both edges of both sides of the cladding, and all these phenomena. Then also we can have some deposition from the cooling water on the cladding, and all these phenomena can lead potentially. I mean, we prevent it, but they can lead to some failures that we need to understand well to prevent them. So what we do at PSI, we have a special FIB, which is placed in a hot lab. So it's a FIB that can also produce small, tiny samples from highly radioactive materials. And then such sample, so we can extract a pillar even from a spent fuel, which is itself very highly active. But when we downsize the sample, then the activity is not so high. And actually, we could even bring it to different beamlines, those which do not have the license for dealing with radioactive materials. And then such pillar, we could place at the sample stage, sample manipulator of the microXAS, and measure different things. So here's, I would like to actually start with the 2D imaging. So this is a thin lamella, also extracted using the FIB of the cladding of the zircaloy, and for which case we performed 2D imaging. So we were scanning in 2D over the area around the crack. So this is this crack in the zircaloy. So this is this zircaloy cladding and on purpose induced crack. And we were studying the distribution of hydrides. So here we could perform this mapping of different types of hydrides. We actually could notice that we have, so based on diffraction contrast, we could see that we have two different types of hydrides, which was before not clear if one of them is actually stable. People believe that it's metastable, but actually we can see that it stays in the structure. We could also, based on fluorescence, see differences between two different parts of the alloy. I am not going into very details of it, but so here we could see that we have two different alloys, and the difference we could see essentially based on fluorescence and diffraction contrast. And thanks to this, we could see that at the edge of these two alloys, we have formation of hydrides in the high concentration. So we could see that these hydrides are agglomerated at this edge, but also at the tip of the crack. So the interesting thing is that the stress induces formation of the hydrides. And then on the other hand, the hydrides are very brittle. So then which accelerates the formation of the crack itself. Yeah, so then before I move forward to the next case, I would like to say something about the 3D imaging. So, because this is actually a direction where microXAS, where our beamline is going towards 3D or even 4D imaging. So 4D meaning a time resolved. So we can do these experiments that I presented already. These types of experiments we can do also in 3D. That means that we can scan the sample in X and Y and also rotate. And then for each, and this is an example of scanning for tomographic slice, for obtaining a tomographic slice in such a pillar. So we scan the sample in X and we rotate the sample. And then for each pixel, essentially, we get the spectra. So in this case, fluorescence spectra for each pixel. And for each rotation, again, another one and another one. From this signal, for each energy bin, we can construct sinograms. And then using tomographic reconstruction algorithms, we can obtain fluorescence contrast images for each energy. And based on this, we can map, and of course, this we can do for several slices or many of them, and then finally obtain the 3D image. If we do it with diffraction, we of course first we do, measure diffraction rings to the images of diffraction rings, which first we need to azimutally integrate. And then the further procedure is the same. So based on the patterns which we have to, for each measured pixel, we construct sinograms, then we perform tomographic reconstruction, and then we can have a 3D image of crystalline phase distribution. And here is an example, again, from spent nuclear fuel. And this is actually what you see here is actually a high resolution imaging based on attenuation. Actually, this one is on phase contrast, but we also have it based on attenuation. So this is a result of phycography performed at CSAC's beamline. And you see that these are two pillars extracted from different parts of the nuclear fuel, spent nuclear fuel. And in this signal, with very high resolution, we could see that we have some inclusions inside or pores or whatever they are. Yeah. However, from this contrast, we couldn't see, we couldn't say what are they. Yeah. We couldn't even say if they were really pores or filled with something. And what we did, we took the same sample to microXAS and we performed this tomographic microscopy based on fluorescence contrast, but also on diffraction contrast. And then we could match exactly same slices from phycography, which gives us very high resolution, with much poorer resolution, but fluorescence contrast. And we could really recognize that in this, that these features that we observed here were filled with krypton. So there are pores filled with fission gases. And then depending on the burnup of the fuel, depending on the position, we can analyze their pressure, concentration, and more things. Yeah, and in fact, we did it really in 3D. And yeah, you can see here the 3D image. In fact, okay, but I will move forward. And another example was, but here I try to go a bit faster. And another example is tomography of the fuel with a piece of cladding. So you see, this is a part, so again, FIP extracted piece of spent nuclear fuel with a piece of cladding. And here are several tomographic slices based on fluorescence contrast. Yes, so this is slice 1, slice 2, slice 3, just like we have it here on this picture. These red lines indicate the positions where the slices were taken. And here we can nicely see how zirconium is intercalating into the uranium oxide. And in fact, yeah, this phase that we see here, which we could then recognize from diffraction contrast, was at zirconium oxide. So we see the zirconium oxide growing into the fuel. So I will move forward. Then another field of many studies performed at microXAS is biological samples. This is a nice, easy to present example of a comparison based on a fluorescence contrast imaging, comparison of some seeds of plants, comparison between wild and mutated seeds. And we could define here that the distribution of zinc is completely different that in the mutated seeds. The zinc is only on the shell, agglomerated only in the shell, while in the wild case, it's in the whole volume. Another interesting example is the, sorry, went too far. My computer is a bit slow. Okay, excuse me. Let me change. Yeah. So this is a Daphnia. Daphnia is a waterfly. This is a little organism that is kind of indicator of the quality of water. And this is, but what is important is that this organism is also like lowest step in the chain, in the natural food chain. And there is a group of our users from Norway. They feed these little animals with all the toxic stuff that they can find, and for instance with uranium. And then here at microXAS we could nicely do the imaging of the whole organism and see where different elements, chemical elements, among others uranium, where do they agglomerate. So if the Daphnia will eat it, will it just go through the whole organism, or will it agglomerate in specific organs and stay there? So that's why this was important. Then another next field that I mentioned that is very important now at microXAS is advanced manufacturing. And we are focused, but not only, on two techniques of so-called 3D printing, so laser powder bed fusion and laser directed energy deposition. So, and we do different types of experiments for this case. So, for instance, here, which was very nice also, actually typical experiment for microXAS beamline is the imaging of chemical distribution of chemical elements at the interface between two different metals. So this is a cross-section of a sample produced with this directed energy directed energy deposition technique. So here we have steel And on top of it, we have several layers of tungsten. This is a very interesting combination of two materials that actually would have application in also in reactors, but in fusion reactors. So we're using fluorescence and diffraction, microimaging. We can see nicely the distribution of these elements at the interface. We can see the mixing, but also we can see formation from the diffraction contrast of intermetallics. And what is also important, we can see nicely the distribution of FCC phase and BCC phase in the steel part. And interestingly, this was the distribution for one technique, and for the other technique, this laser powder bed fusion, we got completely strange distribution of the BCC and FCC structures. But this is very important for the mechanical properties of such materials. And even though it was expected to have pure BCC, so then what we do is also, and which is also important for maybe applications at micro beamlines, thanks to the fact that we have very small beam, we can actually capture these small areas also operando, where we melt with the laser during this 3D printing. The molten volume is very small, and then the volume which is solidifying is also very small. So to see it in situ, we also have to use the microbeam. And this is an example of the operando diffraction experiment during 3D printing. So this is a machine, miniaturized machine for 3D printing. Here is the box with the KB mirrors, which focus the beam exactly here. And this is the powder bed. And here comes the X-ray beam. So we are sitting somewhere here. Here is the diffraction detector. And we scan with a laser. And during scanning with laser, we can track the diffraction pattern with a very high speed, currently up to 40 kilohertz. And we can see how during solidification we have transformation of different phases in steel. We can see what happens if we put a new material on it. And yeah, again, I will not go into details, but just an example. And then another example is this directed energy deposition. So you can see also how we can mount such device in front of the detector. So this is also another laser-based device for 3D printing. And here we also did in situ diffraction. And again, with time, we could see what is happening with the diffraction. So, and now this is a presence. This picture you have seen, but in the presentation of Luke. But what is interesting here, he mentioned it was really impressive how fast they were. And I just wanted to add that this was really crazy, because the guys here, they were actually cutting all the cables. They were not disconnecting anything. They were just, to be faster, everything was just cut and removed super fast. And what will happen now with us, with microXAS? So for us, this upgrade is a big challenge, because actually we are the only beamline that is moving to a different place. So we need to build it completely from scratch. We will get a new undulator. Still, we are not really sure what type of, to be honest, but we hope we will get the new one. And what will change in the new setup? So instead of one pre-focusing beam mirror, which was focusing horizontally and vertically collimating, we will have dynamic focusing in both directions. So as I said, we will also have a different undulator, which and we are aiming for the energy range up to 40 keV, so even higher, significantly higher than up to now, but we stay with this 5, so we still have some overlap with tender beamlines. Then we will have again KB mirrors, but we will get new KB mirrors for the future, and we aim for having well, 200 nanometers, maybe with some extra lenses with zone plates, maybe 50 nanometers. So, as I said, in the spatial resolution, we aim to get this factor of 5, 200, 100. We'll see what we manage. It's pretty clear that we will have a huge gain in flux. So therefore, we aim for more time-resolved studies, higher energy, and many new configurations. And then very quickly, I want to go through a few examples which Thomas Hoodwelker shared with me. He's a beamline scientist at Phoenix Beamline, which is similar but operates at a quite tender compared to microXAS energy range. So they also use focus spotlight and scanning imaging. However, they are more focused on spectroscopy, not so much diffraction as we do. So at microXAS, we also do many studies, as I said before, on batteries, but this is also an example that they did at Phoenix. So they prepared a little cell for in operando or in situ studies of batteries. And what was interesting, so they were studying a battery which was believed to that the cycling during charge and discharge is mostly based on a mechanism in redox between iron 2 and 0 redox reaction. But however, this was not well explained just with this reaction. And at this beamline, they noticed that during charge and discharge, also the spectrum of the absorption spectroscopy, XAS spectrum of phosphorus is changing during charge and discharge. And this is, of course, phosphorus is out of the reach for microXAS, while for them it was possible. Another interesting study at Phoenix was studies of calcifications in breast cancer. So they noticed that only healthy tissue can have extra phase called white locite, a phase containing magnesium, phosphorus, and calcium. And this is again, these are elements that we cannot really study the fluorescence of it or spectroscopy of this. So also using actually diffraction wax, they could image and see the distribution of this white locite into several different tissues, and only the healthy tissue could have this phase, which indicates that maybe this could be also a way how to very early predict the formation of cancer in tissues. Another famous example from this beamline was the studies of the oldest carpet in the world, where they did fluorescence contrast mapping and fluorescence of aluminum. So there is a technique well known, but it was believed to be a few hundred years old, this technique of fermentation of fibers to dye them. But it turned out that, and this fermentation allows for the pigment to go inside the fiber. And the pigment contains aluminum. So the point was that they did this mapping of aluminum in this, in the fermented fibers, also fibers extracted from this carpet. And they noticed that they have aluminum also in these fibers inside the fiber, while in the natural case, unrefined fibers, the aluminum is only outside. Therefore, they concluded that this technique was known already 2,000 years earlier than people believed. And then that's the last example of the that was also quite famous study at Phoenix of the Stradivari or other very famous, extremely expensive violin instruments. So there was kind of always a mystery why specifically these instruments are so great. And at Phoenix, they could see that actually the pieces, the wood, is soaked, contains some amount of aluminium, which suggests that some special treatment of the wood by Stradivari, before constructing these instruments. So again, this is a spectroscopy on an element that we could hardly access at microXAS. So here I'd like to thank you, and I'm open for questions.
Małgorzata, thank you very much for this very interesting presentation showing enormous research potential of microXAS beamline. Actually, we are running out of time, but maybe if there is one question, you could answer it. Let's have a look.
Okay, if no question, I just wanted to make a remark that actually for me, I didn't realize before that you do experiment in open air.
Yes, we actually do.
Not in some vacuum chamber or something like this, but this system looks so open. I mean, it is just in reality also. It is somehow.
No. Our sample is for ex situ experiments at least is placed indeed in air, and only for cases, if we try to push towards, for instance, aluminium, then we had some systems of blowing with helium on the sample. Still, it was open. So in this energy range, it's possible. While if you go lower, then it would be problematic, yes.
This changes of this last mirrors, focusing mirrors, how you do it? Is just some switching stuff, or you do it each time by hand?
So you mean the KB mirrors for focusing of the beam?
To change focus.
So that's actually quite a drawback at our, well, I wouldn't say drawback, but yeah, that's a bit a thing to do at our beamline, that indeed every experiment requires refocusing. Every time we change energy, we need to focus again. And we do it essentially manually. We have actuators on the mirrors, on the benders, and we just manually scan over the applied force, so to say, of and see what gives the best focus possible. And it's kind of art, So it's also kind of competition at the beamline, who can focus better. But we need to do it every time, for every experiment, indeed.
Okay, I can see there is one more question from Galina Dovbeshko.
So, Małgorzata, thank you very much for a very interesting report. And my question is about what low limit of concentration of metals can you detect in biological samples?
So, of course, that depends on the sample. So essentially it's ppms, single ppms.
We can detect ppm, yes.
Yes, but then it also depends on the sample. What will be because we are talking about fluorescence, and then what will be the self-absorption of the fluorescence in the sample? How big is the sample? Does it manage the fluorescence, emitted fluorescence doesn't manage to get out from the sample? So this is always a question. But if the geometry is perfect, no issues with self-absorption, then it's single ppms. Yes.
Okay, thank you. And really, is it possible to apply to you with this problem?
Of course, but in 2026, because now we have this big upgrade, which will last until late of 25. However, our beamline is in the last phase of upgrade, and our beamline will be available for user program maybe in the mid of 26. But if you have any questions, feel free to contact me. We can also, I could also suggest other beamlines at different synchrotrons. Yeah, if there is a hurry.
Okay, thank you very much. And I suggest if you have some more questions, please write them in a group chat. And Małgorzata, I will ask you to answer them if some appear. Okay, and we have to move on to the next speaker. So Małgorzata, thank you very much once again for the great presentation.